lab that I work in, the Grant lab, studies endocytosis, specifically receptor-mediated endocytosis. So endocytosis is the process by which cells take in molecules from the outside. So obviously this is really important for cells taking up drugs and nutrients and numerous other processes that are vital to cell life. So our lab studies all these different proteins that are important in the process of endocytosis and maintaining the different compartments within the cells that regulate endocytosis. My project is actually focused on a particular group of proteins called snare proteins, and a lot is known about snare proteins in both yeast and mammalian cells, for instance. However, quite little is known about snares in C. elegans. So having the knowledge about how particular snares function and what their localization is in C. elegans cells would be really useful to have as a tool to further study endocytosis in C. elegans. And C. elegans is this really small nematode worm. Adults are only about 1 millimeter in length. And if you go down to the Raritan River and you dig up some soil, you'll find C. elegans crawling around in it. So it's a really common organism across the country, across the world. And it's a really common organism to use to study cell biology, genetics, and other biological functions because it's easy to maintain. It's much easier to maintain, say, than fruit flies or mice, certainly. Because you can freeze worm strains, and there are other advantages such as that. In addition, all of the biology that we've found in human cells that regulate endocytosis is conserved in C. elegans. So that means all of the proteins that we know act in our bodies to regulate how cells take in molecules from the outside, they have homologous proteins in C. elegans. In addition, C. elegans is transparent, which is really useful because we can visualize abnormal structures in the worm, and we can also visualize the localization of fluorescent proteins in the worm, so where those fluorescent proteins localize to in cells. The goal of the research really was to further characterize this group of proteins in C. elegans. So we really didn't know barely anything at all about these proteins in C. elegans. So we wanted to understand where they localized to in the cell and what their function was. So we were able to create hypotheses about where every single snare in the C. elegans genome localizes to based off of our production of the worm strains that expressed the green fluorescent protein linked to the snare protein. The professor that I've worked with while at Rutgers, Dr. Grant, has been incredibly supportive and has been a really great mentor to me by helping me to figure out how should I approach this problem, what kinds of experiments can I do, maybe I should take a look at this other paper that has some interesting idea in it and then we can discuss it. My experience at Rutgers has kind of given me that appreciation for developing your own hypotheses, for designing your own experiments, and for carrying that process through. And it's something that I've enjoyed an incredible amount, and is something that I definitely want to continue in my career.